Hello students, myself Popere KS, Assistant Professor in Botany at Shikshan Sanstha's RBNB College, Sri Rampur. In this lecture, we are going to discuss about the topic Endoplasmic Reticulum, or this is also called as ER. So, this Endoplasmic Reticulum, the term means, endo means inside, so endo means inside, plasmic means cytoplasm, and reticulum means the network. So it is a network of membranous system or membranous structure that are found in the cytoplasm. Now this was first observed by scientist Garnier, and the term endoplasmic reticulum, it was first given by a scientist called Popere. So he has observed the endoplasmic reticulum first, and the term first was given by the scientist Popere. Now next to that, the endoplasmic reticulum, it will be present in all the eukaryotic cell and it is absent in prokaryotic cell. So prokaryotic cell, the endoplasmic reticulum, it is absent, and in eukaryotic cell it is present, except in mature RBCs. So exception is so this ER will be present in all the eukaryotic cell. Exception are there except the mature RBCs. The mature RBC, they don't have presence of endoplasmic reticulum. Even the egg cell, embryonic cell or germinal cell, in this cells the endoplasmic reticulum, it is absent. Now we will see the morphology of the endoplasmic reticulum, or the that is the element of endoplasmic reticulum. So this ER mostly they will be present surrounding the nucleus. So suppose so this is the endoplasmic reticulum. It is in continuation with the nuclear membrane. So this is nucleus, this is nuclear membrane, and in continuation with the nuclear membrane, that is the nucleoplasm, there is presence of ER, or the endoplasmic reticulum. Now this ER, it consists of the different part. So first is cisternae. Now what is cisternae? Cisternae, these are the sac-like structure. These infoldings, these are unbranched These are sac-like folding or membranous system. And the lumen of this cisternae is from 40 to 50 μm. They will be in closer to the plasma, that is the nuclear membrane. So it is in close to the, they are closer to the nuclear membrane, this outer nuclear envelope. And next structure is the vesicle. Now this vesicle, they are present So these are the vesicle, these are the structure which will be present in association to the cisternae. So they are present on the cisternae. They are spherical structure that is also from 40 to 50 μm. So their lumen is 40 to 50 μm. These are also unbranched structures. Now after the vesicle, the third structure is the tubule. Now the tubule is this region. This region is called as the tubule. So this is tubule. This is cisternae. Now this tubule, this is branch structure and their lumen, or they are means it has a larger surface area. It will be from 50 to 100 μm. They will be close to the, that is to the plasma membrane. So this cisternae, they are close to the nuclear membrane. The tubules, they are closer to the plasma membrane. They are branch structures, and on this cisternae and vesicle, they have presence of ribosome. So they are studded with the ribosome. So on cisternae and even on the vesicle, there are presence of the structure. These are the ribosomes. But on tubule, the ribosomes are absent, or ribosomes are not present on the the tubule. So this is about the morphology or the element of the endoplasmic reticulum. Now on the basis of the presence and absence of ribosome, the endoplasmic reticulum, they can be divided into two. So two types of ER. So first one is RER, or it is also called as granular ER. RER stands for rough endoplasmic reticulum. Now why they are called as rough endoplasmic reticulum? Because they have presence of ribosomes. So on the rough endoplasmic reticulum, there are ribosomes. They are studded with ribosome. Now on this ribosome, they are present on a transmembrane glycoprotein, that is ribofurine. So on the endoplasmic reticulum, on the ER transmembrane, there are presence of glycoprotein. This glycoprotein are ribofurine 1 and 2, and on this glycoprotein, the ribosomes are present. Now during the protein synthesis process, as the polypeptide chain which is synthesizing on the ribosome, it will enter into the ER lumen through the channel which are present on the ER membrane. So through the ER membrane it will enter into the ER lumen. So it will, the polypeptide enters the ER lumen. So after that what will happen, there will be the processing of the polypeptide will takes place, the folding will takes place. So this processing, folding and the maturation of the polypeptide will take place in the ER lumen, and later on when then it become functional, so then it is called as functional or protein. Now it become functional protein. So after that what will happen, now from the rough RER there will be a pinching of a vesicle. A vesicle will pinch out and it will fuses with the Golgi. So this is how the protein synthesis process will occur in the ER. Now there is also some function which will be occurring in the RER, that is synthesis of the membrane protein, that is the glycoprotein. So glycoprotein synthesis will also take place in the rough endoplasmic reticulum, and also one process, that is biogenesis of cell membrane. So this will also takes place in rough endoplasmic reticulum. Now next is the smooth endoplasmic reticulum. In smooth endoplasmic reticulum, now here ribosomes are absent, so there is no presence of the ribosome. Now here what will happen, here lipid synthesis, phospholipid synthesis and sterol synthesis will occur. So these three processes, that is the lipid, phospholipid and sterol synthesis will take place. Here also the metabolism of carbohydrate takes place. And the toxic detoxification. So toxin detoxification will also take place in smooth endoplasmic reticulum. In muscles, it will this will also regulate in muscles, it will also regulate the calcium ion concentration. So this due to this there will also induction of the motor impulse in case of the muscles. They are found in variety of plant and animal cell, and it will also help in a process, that is gluconeogenesis. So what is this gluconeogenesis? It is a process in which in which what will happen, glucose 6-phosphate, it is converted into glucose in presence of an enzyme glucose 6-phosphatase. So in case of SER, glucose 6-phosphatase enzyme is present in smooth endoplasmic reticulum. So the gluconeogenesis or the glucose 6-phosphate, it is converted into glucose. Now this is about the rough endoplasmic reticulum and smooth endoplasmic reticulum. Now we will see the functions of endoplasmic reticulum. Prior to function, let us discuss the chemical composition. So chemical composition, mostly the endoplasmic reticulum, it will have presence of ribosomes, this ribosome, they consist of ribosomal rRNA, and in case of the eukaryote, the ribosome is 80S. In case of prokaryote, it is 70S. That is 40S plus 60S. Next, so this is 30. Eukaryotic cell, that is 80S, 40 plus 60S, 70S in case of the prokaryote, that is 30 plus 50S. So this S is the Sedgwick unit, or it is called as the sedimentation coefficient. This S or the Sedgwick unit, these are not additive. Now, so this is about the ribosome which are present on the ER. Now next is one more thing, that is protein. They are present on the they are present in the endoplasmic reticulum. Now for the protein synthesis, different rRNA are required, that is ribosomal rRNA, mitochondrial mRNA and tRNA, that is rRNA, mRNA and tRNA. Now rRNA, these are the ribosomal RNA. This will be present in ribosomes. They are important for the protein synthesis process. Now next is the mRNA. This mRNA, they will be synthesized in the nucleus and will transport it to the cytoplasm, and they are involved in the protein synthesis process. By using the codon which are present on the mRNA, protein synthesis will occur, and this tRNA, this will do the function of, or it will brings about the amino acid during to the protein synthesis machinery during the translation process. So these three are important for the protein synthesis process. So they will be present on the endoplasmic reticulum during the protein synthesis process. So after that, the chemical composition. Now let us see the functions. So the functions of endoplasmic reticulum, first is mechanical support. So this endoplasmic reticulum, it will be present in the cytoplasm, or it will divide the cytosolic fluid. So it is working as a cytoskeleton of the cell. So it is giving mechanical support to the cell. Next to that, next is protein synthesis. So on ER there are presence of the ribosome. Ribosomes are called as a protein factory. So with the help of the ribosome, mRNA, tRNA, protein synthesis will occur in the endoplasmic reticulum. Also the maturation of the protein will also take place in the ER lumen. Next to that, then the transport. Transport, that is the import and the export, and also the intracellular circulation of the different protein, lipids or the enzyme, it will take place in the endoplasmic reticulum. Next is biosynthesis of the lipid. So the synthesis and even the storage of the lipid, it will take place in the endoplasmic reticulum, and particularly in the smooth endoplasmic reticulum. Last is oil body biogenesis. So in the endoplasmic reticulum of the plants, there are different enzymes and the structural protein which are involved in the oil body biogenesis and also in the lipid storage. So these are about the functions of the endoplasmic reticulum. Thank you.